Hello, this video going to be about population genetics, breeding, selection and fitness. And here's a problem. The following data were collected from a population of Drosophila segregating sepia and wild-type eye colors. A sample was taken when the eggs were deposited and later among adults. Reconstruct the model of selection. And if you know how to solve this problem, I recommend you to stop video here, try to solve this problem on your own first, and then when you would be ready you can run video again and you can compare your answer with my answer and explanation. And before I will explain how to solve this problem, I just want to remind you that this color, sepia, is brownish red, and a wild type is just black color. So here we have, if you take a look at this table, three genotypes. This genotype would be black color. This also would be black because wild type is always dominant to the other types, and this type would be sepia. So this is going to be reddish brown color, and these two would be black. So this is going to be heterozygous, this is going to be homozygous, and this one also going to be homozygous recessive. And now we can take a close look at this table here. So what we see is that when eggs were deposited, the number were 25, and here we have 50 and 25, so total number is 100. So if we add all these numbers, we are going to get 100. So it's very easy to say the frequency, that this genotype is 25% and this genotype 50% and this genotype 25% of the total deposit eggs. But when this Drosophila become adult and we count once again 100 adults, so if we add all these numbers we are going to get 100 once again, and we find the different frequencies here. So this genotype, instead of 25%, now would be 30%. This genotype also would increase in frequency, so would be 60% instead of 50% initial. And this genotype, that is homozygous recessive, would represent only 10% instead of 25 initial percent. So we see increase in frequency of this genotype and this genotype, and decrease in frequency of this genotype. So this is due to different fitness. So that means that this genotype and this genotype have better fitness than this genotype. So this also means that not all specimens of this genotype would survive until adulthood. So now let's find what is the fitness of each genotype and phenotype. So in order to do this, we have to divide, for example, in order to find the fitness here, we have to divide 30 by 25, and we are going to get 1.2, and this is going to be fitness of this genotype. The same thing we can do here, and we have to divide 60 by 50, and once again we are going to get 1.2. So the fitness of these two phenotypes, or this is going to be one phenotype, these two genotypes would be the same. And now we can calculate the same fitness for the last genotype, and we have to divide 10 by 25, and as you see the fitness here would be 0.4. So as you see, the fitness of this genotype would be much less than these two genotypes. So, but this would be absolute numbers, and in order to work with scientific data, we need to bring these numbers on the scale between 0 and 1. So we call this relative fitness. And in order to find relative fitness, we just have to use the fitness of the fittest genotype. And here we have two such genotypes, not one. So we would use this number 1.2. So if we divide 1.2 by 1.2, we are going to get 1. And the same here, we are going to get 1. So this is going to be relative fitness. And here we also have to divide this frequency of this genotype or fitness of this genotype by the fitness of the fittest genotype. So we have to divide 0.4 by 1.2, and the relative fitness of this genotype would be 0.33. And what all these numbers tell us? So this is going to be our answer. This is going to be relative fitness of each genotype, and what this answer gives us. And using this information, we can say that relative fitness of this genotype would be much smaller than these two, and we are going to lose at the end this allele, S allele, and after certain number of generations we are going to lose it completely, and there are going to be fixation of this S plus allele. So this allele would be gone completely, and even if we would see this allele in a population, this is going to be only due to mutations, and the number would be very, very small. And this is all for today. Thank you for your attention. Please subscribe for my new videos that I post almost every day. Thumbs up if you like this video. Please write your comments, questions if you have any. And see you in the next video. Goodbye.